Hi everyone, my name is Braden Bell, and today I'm going to be taking you through the protocol for using ImageJ for DNA damage analysis. So ImageJ, as you probably know, is a program that analyzes images in a bunch of different ways, but today we'll be using it to measure how much of our fluorescent marker is present in our cells after our treatments. So before we begin, a few things. You're going to want to download ImageJ or FIJI, they're the same thing. And you're going to download JMP or Excel, depending which one you want to use for statistical analysis. You're also going to want to download our two macros we'll be using today, which are the ZStack Builder and Analysis macro. Your instructor will direct you to the download location for those macros. And you're also going to have the images downloaded onto your laptop or computer that were taken with the confocal microscope. And these images should be in a .laf format. I'll show you that a little bit later. And then we will get into making some folders. It's really important to be organized in this protocol. Things get crazy very quickly. So I have everything organized in one folder called ImageJ. I have my two macros, my analysis macro and my ZStack Builder macro. Here's FIJI or ImageJ. And then I have my one treatment folder containing all of my other stuff. So this treatment we treated ourselves with siRNAs targeting SPOTA5, which is a novel gene involved in cell cycle and proliferation. And so you could have one treatment like this one, or you could have two or three. If you do a whole slide by yourself, you could have 8, but for today, we're just going to do 1. And so let's look what's inside there. So inside our this SPOTA5 siRNA file, we will have our image with the .laf format. This is the one that's taken the microscope or the one that's provided for you. I would organize it like this. And then inside this folder, in addition to the .laf folder, we have a folder, empty one, called DAPI and one called H2AX. It is important to have empty folders labeled like this, or for whatever markers you're using to use this protocol, just so our data goes to the right place for each treatment. So important to have these. And yeah, now that we're set up, we can actually get into analyzing some data. Okay, so now's the time to open ImageJ. Take a second to load up. I'm gonna X out of this. And on Mac, and I think on Windows, I am on Mac just in case anyone didn't notice. So this might be a little different if you're not using a Mac, but a lot of it's the same. So this little bar, this is ImageJ, this little bar that pops up. So basically, there's gonna be some updates. There might be updates to the program. You're gonna want to download those before lab. But yeah, anyway, first we're going to do file open, and then we're going to go to our two macros, and we're going to open them both up, and this should open up with this sort of dialog box. And file. Also for Mac users, it gets a little interesting. You have to click on the menu and then do file open. For Windows users, the bars are just up here. So keep that in mind. Make sure you click on the bar before you do anything, otherwise it'll mess it up. Then we're gonna go to ZStack Builder and open that one. And now we have our two macros. You can flip back and forth between them over here. So that's all good. We're gonna start with our ZStack Builder. So this macro is up and open. I wouldn't mess with any of this. That would be not great. So now we are going to select the bar and do file open. And now we're going to open our .laf file from our treatment group for the siRNA targeting SPOTA5. So it's going to say up here, we're reading headers, and then this menu is going to pop up. Basically, I wouldn't mess with any of this right now. You shouldn't change any of these options, but make sure open all series is checked and the color mode is grayscale. You shouldn't have to change anything, but if these aren't checked, make sure they're checked. And then we're going to click OK. And so this console will pop up. It looks big and scary, but it's not really important right now. And so eventually our image will pop up, but it'll take a while. There it goes. And so it's going to start opening all of the images we took in the .lif file and bring them up to our screen. So I'll come back to you when those are ready. All right, so now all of our .lif files have loaded. As you can see, we have 34 images. As you can see by series 034. So these are they're basically 20 images, well, 40, laid on top of each other. So it's almost like a 3D image as we can scroll through. So this is good and interesting. So we can see like different levels of the cell. And also we can see our separate channels. It's like you can see some DNA damage in this cell down here. That seems to be the only one. Anyway, but basically to analyze these images, we need all of these Z stacks is what they're called. We need all of them to become one image. So to do that, that's why we have our Z stack builder macro. And basically you are just going to, click the first in the stack and you're going to hit run down here. And then you're going to want to put a file name in front of these. So for me, I'm going to put my initials and then what my treatment is and just hit OK. And then we're going to sit back and it's going to flash through all these boxes. It's going to convert them to the file type we need and then rename them. And we're just going to wait till that's done. Now that we've created our Z stacks, we're going to click this menu bar again, do File, Open. And as you can see in our SI Spotify file, we have a lot of new images. So basically, the Z-Stack Builder macro took our LIF file from the microscope, broke apart the individual images, and then combined them, the 40 images into 2 images. Since we have 2 channels, there's 2 images for each channel, or 1 image for each channel, 2 images total. And so now we have 34 separate images that are all different, and they're ready to be analyzed. So we're going to open the first one because that's how the program runs. And now we are going to, since we have this open, we're going to make sure we're on the analysis macro and we're going to hit run. So it's going to ask you to open or to make sure the file series 001 in the name is open, and it is, so we're going to press OK. How many channels are present in the sample? Usually it's going to be 2. The default's 2, but you might have more. Then it's going to ask if there are empty folders in the ZStack folder and are named the same as following entries. So if I open this, you can see all of our new .tif files. And we also have DAPI and H2AX folders that are empty. And this is in our treatment, so this is good. So X out of that. And then we are going to click OK. And our channel 1 is DAPI. That's what I use for channel 1 is DAPI. And I have an empty DAPI folder. And then my channel 2 is H2AX, which measures DNA damage, as you know. And that was my second empty folder. So I'm going to click OK. So then it's going to ask what image I want to start at. So sometimes, occasionally, this program will crash or there'll be an error, or your computer dies or something. This is a good way to like, oh, I stopped at image 13 before it crashed, so I can go back to image 13 if I wanted to. But for just the sake of starting, we're going to start at image 1. So then it's going to do some flashing. And now we're finally going to be able to select our cells. So your instructor will probably talk a little bit more about what cells are good cells to analyze and what cells are not great examples. There's also some examples in the protocol if you're looking for more examples. And so basically, this is a like 0 or 1 situation. So if you want to keep the cell, you're going to keep the 0 and just press enter. So I'm going to keep this one. And then it's going to delete the cell, which means it was selected. And then say, okay, like cell number 2 is now highlighted in blue. So do I want to keep this one? 1 would be no, 0 would be yes. So I'm going to keep this one too. I'll keep 3, I'll keep 4 and 5. I'm actually going to, I realize I'm going to keep all these, so I'm just going to press enter. And so now it's going to save them, close out. Actually, it's going to pop up this menu, just click discard. And it's going to say, okay, the next image coming up is image 2. So we're going to click OK. And now we have our second image. So now, same thing, go through, select what we want. All these are looking pretty good. I'm not sure about 7. Just because I'm not sure, I'm going to put in a 1 for no. And as you can see, it just deselected it and didn't delete it. So if it deletes the cell, it was successful and it was selected. And if it doesn't delete it, it wasn't selected. So the rest of these look pretty good. So I'm just going to go through those. Same thing, discard, and it's going to close everything. And then the next image coming up is 3. And then you basically just go through this until you have finished analyzing all of your images. So like there's 34 for this one. I'm not going to go through all 34, but I'm going to do a few more for some more examples. I'm going to say no for this one. I'm a little interested about what's going on here. So I'm just going to do no. Do no for this one, that one. If you have a lot of images, you can afford to leave out some cells that might not be the best nuclei to analyze because DAPI identifies nuclei. So that's up to your discretion and up to your professors what they want as well. So go through this one as well. 3, 4, 5 looks good. 6, 7 looks pretty good. 8, I'll do 9. And then 10, 11, 12, those all look pretty good. These are some good images. Discard those. And then on to image number 5. This will be the last one I do. No on that one. I'm being really picky. Usually, you don't have to be this picky. It usually does pretty good. And so I don't want to, I'll finish at that one. And then now we'll get on to getting the data into somewhere where we can actually analyze it. Okay, so I've closed out of ImageJ or FIJI. I'm going to go back to my folder. And you can notice in my Spotify folder that I made, I have my .laf, my .tif, but now in my once empty DAPI H2AX, I have these CSV files. And basically, these are like Excel spreadsheets. They're numbers, and we can import them to JMP or Excel, depending what you want to use. I'm going to use JMP, and I'm just going to import them directly to JMP. I know the protocol might say something different, but this might be the best way to do it. That's what I recommend. So I'm going to exit out of my Finder. I'm going to open JMP. I'm going to go to File, Open Multiple. Then click this little button. I'm going to go to Desktop, ImageJ, Quantify, and I'm going to open my DAPI folder. Open, import. So then this comes up. Perfect. These are my DAPI results. I'm going to go File, open multiple again, and then do the same thing, but with my H2AX. And import. So now I have 2 separate sheets for DAPI and then one for H2AX. You can ignore this one back here. I want to combine these. I'm sure there's a really fancy way to combine these. I'm not super proficient in JMP, so I'm just gonna copy and paste because that's easiest. Maybe. Okay, so I have this side over here is DAPI. Make sure you don't forget that. This side is DAPI, and this side is Gamma H2AX. So this is all of this treatment is our SPOTA5, SI SPOTA5 treatment. And if you were analyzing multiple, which I will probably load in after I finish showing you what's going on here, you'll want to keep everything in one big JMP file, just because it's better for comparisons that way. So you're going to add a treatment category. Like this treatment was siRNA targeting SPOTA5. So you're going to record that for all of these data rows. But maybe we want to add like a negative control, like an untreated value. So we'd add more data from a different image set and then put untreated in our treatment category. Or maybe we want to do a positive control like actinomycin D, which is a DNA damage causing drug. We'll put that in the treatment category or column next to the data that corresponds to it. But for right now, this is just one treatment. So to check that you've imported everything right and everything matches up, basically we want to make sure that we're comparing the DAPI values of a cell to its own gamma H2AX values. So we don't want to like get mismatched where like we measured the area of one cell, but then its fluorescence was the value of a different cell. So basically, on the left, we have the DAPI channel. On the right, we have the ImageJ channel. The size of the cell should not change from channel to channel. So right here, the area of cell 10, or nuclei 10, I guess, not cells, these are nuclei, was 204.79. And if I look over here, this one is area, the labels got lost when I copy and pasted it, but it's fine. The area is also 204.79, so those match up, which is good. That means this cell had a DAPI intensity of 88,000-ish, and this one had a gamma-H2X intensity of 2,000-ish. So going down, I see the area of this one is 212.26, matches up to 212.26, so it looks like everything's good here. So I'm going to go add in more treatments just for comparison and then analyzing them and building a graph. But this is the basics for now. Okay, so I imported a new file. We have some more treatment groups like Spotify L1. These are still siRNAs, so pretty similar to what we're doing for DNA damage. You might do plasmids and overexpress the genes to see how that affects DNA damage, maybe not. So we have H2AX signal right here, raw integrated density, which is this column, and we have DAP integrated density, which is that column. Mostly what we're worried about is this H2AX integrated density, because that measures DNA damage. Once we matched up the two sets of data, the areas, to make sure like everything matched up fine, all is good. If you wanted to compare it to the DAPI, you'd have to do that. Right now, we're just going to look at integrated density for your H2AX. And when I analyze this, I want to normalize it to something. And so I like to normalize it to the average of the treatment groups. So like, or not the average of the treatment groups, I'm sorry, the average of the untreated value. So I would take the average for my untreated treatment and then divide all of my H2AX values by that average. And then that'll give me this new column. And so, okay, so to normalize this data, I'm going to go to analyze fit by y by x. I'm sure there's an easy way to do this, but this is the way I know how to do. So I'm going to do that. Take my integrated density of H2AX, my treatment group. Then I'm going to go to means and standard deviation. I'm going to find the mean for my untreated value, which is here, which is 12,904.705. So I'm going to save that number. So that's the average DNA damage for my untreated. So I'm going to do that, keep that. I'm going to go to this column, do formula, and I'm going to take my H2AX raw integrated density column, and then I'm going to normalize that by dividing it by the average of the untreated, which was 12,904.705. Enter, then hit OK. Then we should get this value over here. I'm going to rename it. Oops, 2x. Okay, so click OK. And now we can get into our analysis methods in JUMP. You could do a fit y by x with our normalized, and then factored by the treatment. And then we could do like a student's t-test and look at significance. So basically we're seeing here is compared to the untreated, our treatments for SI Spotify L1 and SI C1RF109 are significantly higher than our untreated values, normalized wise. And we could probably see that if we go to graph builder and then we do normalized gamma H2AX integrated density. I'll put this up here by our treatment. So as you can see, we definitely have, here's our untreated group, we definitely have some sort of increased amount of DNA damage compared to that value. These ones don't seem to have a significant amount of DNA damage, which we saw in our student's t-test, but you can kind of see that there's a excess in these two treatments. So yeah, that is in short how you would use ImageJ to analyze these cell nuclei, quantify them, and then use JUMP, or not Excel in this case, but use JUMP to analyze the resulting data. So hope you enjoyed. Reach out to your instructor if you have any questions, and refer to the protocol. Thank you.